In this chapter, we're going to go over both uncertainties and errors in measurements. So the thing to be aware of is that every time you take a measurement, there is going to be some uncertainty and some error associated with that measurement. You can never be completely certain of a result of a measurement. So an example of this would be what is high blood count. So if today's blood count is 5.6 million cells per microliter, how do you know that that is a normal blood cell count? Well, you would have to know what normal means. So on days when you were healthy, so there's no underlying conditions that you have, you're not sick, you're not experiencing anything unusual, you would have your blood count done then. And then notice that we have a number of different results for those normal days. Okay, so this is indicative of error and uncertainty in a measurement. Your condition probably isn't changing, but because the measurement itself has some uncertainty, the result that you get out of your measurement is going to be slightly different every time you do it. Even if it's the exact same sample, it's likely that the result is not going to be exactly the same. So if today's count was 5.6 million cells, your normal count goes from 4.8 to 5.4 million cells, then you would have to determine whether or not 5.6 million cells is a statistical error or is within the normal variation of your blood count. So how are measurements taken and how can we be confident that our result is correct? So if I were to ask you a question, what is the mass of a penny? Now that's kind of an ambiguous question because what am I really talking about? Am I talking about, say, a particular penny that I'm holding? What is the mass of this penny? Am I talking about the average mass of all the pennies in circulation? Am I talking about the average mass of American or Canadian pennies? There's a little bit more that has to be built into that question in order to understand what type of measurement needs to be taken. So if we assume it's the mass of an average US penny, then the question is, how do we go about measuring that? Now, clearly you can't take every single penny in circulation, weigh them all, and then average the, billions of pennies that are out there, right? You would have to take a representative sample, determine the average mass of that representative sample, and then apply some statistics, which we'll go over in a later chapter, to determine what the mass is, plus or minus some uncertainty. So some of the ways that we could characterize a measurement are by its mean or its average, its median, the range, standard deviation, and variance. So mean is something that you use quite often. It's just the average of a set of results. It's a measure of central tendency. But one of the problems with a mean or an average is that an outlier, or a measurement that is quite a bit away or different from the rest of your measurements, can greatly influence the mean. So for example, if we had 3 pennies, 2 pennies were very close in mass, about 3.5 grams, and then another penny was much higher in mass, say around 4 grams, that would skew the average mass of a penny towards the 4 rather than keeping it closer to the 3.5, which might be what the actual average mass of a penny is. Maybe the one that's 4 grams has a lot of dirt on it or some other contamination that gives it that extra mass. So the median is the middle value when the data is ordered from smallest to largest, and it is less affected by outliers, since whether or not the outlier is a high value for the measurement or low value for the measurement, the middle value is still going to be probably somewhere in the range of where most of the measurements are. So most of the values will be same around the middle, and so the outliers are not going to affect it so much. So you can practice with the example data set below if you like, but both mean and median are pretty common and pretty easily done. And in lab and in most of this class, the average is going to be what we're going to be using. So another way to measure the uncertainty in a measurement is to measure the spread, or the differences in the values of individual measurements, so the difference between the highest value and the lowest value. So we can measure that in terms of a range, which is simply that difference, maximum minus minimum, seeing what that range is. But a more useful measurement is the standard deviation, which gives you the spread of the data about the average. Okay, and a smaller standard deviation means less variability, which would translate into greater precision or greater reproducibility. More measures of spread, relative standard deviation, is something that you will be doing in lab, or you have already done if you've already done the KHP lab. So the relative standard deviation, or SR, or RSD, is the standard deviation, which is S, and then X bar, that is just the mean of your measurements. So relative standard deviation would give you a number less than 1. A percent relative standard deviation would be relative standard deviation times 100, and in lab you're asked to do a relative standard deviation in parts per thousand. So instead of multiplying by 100, you would just multiply the relative standard deviation by 1,000. Variance is something that we're not going to use in this class at all, but it is another measure of spread that may be used in other courses that you take. So the point in taking any measurement is to try and figure out what is in the sample that you are looking at. So you're asked to find a concentration of something. You want to be certain that the concentration that you get from your analysis is what is actually present in the sample itself. So do the measurements represent an expected result? Maybe you don't know exactly what that expected result is, but you might have an idea of what your answer should be based on past analyses or the nature of the sample itself. Okay, so are the measurements accurate? That's going to be something that's going to be important. And the question you want to ask, maybe why is there variability in a result? Okay, are the measurements precise? Remember, if a measurement is precise, there's going to be very little variation in a measurement. If a measurement or a set of measurements has lots of variability or a large range or a large standard deviation, that means that the precision is not very good. So as I said before, all measurements contain experimental error, and there's no way to get around that, but we can take it into account. And the next couple chapters we'll be looking at how we can do that. So what kind of errors are there? So let's just talk about them specifically now. So we have systematic errors and random errors. Okay, again, nobody knows the true answer, but sometimes we have an idea of what the true answer could be and maybe what it can't be. For example, there is no way that John Smith is pregnant. It doesn't really matter what his urine sample says. It's just not possible. So there's some mistake in that analysis. So if we're talking about systematic error, another name for that is determinant error. So it can be a positive error or a negative error, meaning that it's greater than the actual value of the sample or lower than the actual value in sample. So if we're talking about concentration, a positive error would mean the concentration you get from the analysis is higher. A negative error would mean that the concentration is lower than what's actually in the sample. So the thing about a systematic error is it's consistent. So usually if it's positive, it's always going to be positive, or if it's negative, it's always going to be negative. And these types of errors can be found and they can be corrected. Some things that can contribute to systematic errors are sampling, how the sample was collected, where it was collected from, the method that was used to analyze the samples, measurement and personal errors. Maybe the pH probe that you're using was not calibrated correctly, so it's always given a pH that is higher than it should be, for example. So we can find these errors, we can correct them, and they can be detected by analyzing what are called standard reference materials. So these are materials that are developed by governments and other agencies that have known concentrations of analytes that you might be looking for in similar matrices. So for example, if you're looking for copper in a lake water sample, there's a standard reference material that has several different types of metals in water, which represents or mimics what a river water sample would be. Analyzing blanks is another way to find systematic errors. because if your blank is giving you a result that's higher than you expect or lower than you expect, then there's something going on with your method. Maybe there's some contamination being introduced in the method. You can use different methods that measure the same thing and compare. The result that you get should be independent of the method, right? Meaning that if you use 2 or 3 or 4 different methods to measure the same thing, they should all give you the same answer because the sample itself isn't changing. So the method itself is being used to determine what's in the sample, so different methods should give you the same result. If they don't, then 1 or 2 of those methods may have some kind of bias, some kind of mistake, some kind of contamination, or some other problem that may be contributing to the error. And round-robin experiments means that one lab would do the analysis, another lab would do the analysis on the same sample, and then maybe a third lab would do an analysis on the same sample again. Maybe they use slightly different methods, maybe they use the same methods, but again, since the sample is the same, each of those labs should give the same answer. And if they don't, then maybe one of those labs has an issue that they have to deal with. So here's an example of a systematic error. So this is showing the actual amount of water that was dispensed from a burette by weighing the water that was dispensed. So for example, if you were to dispense what you think is 10 milliliters, so you dispense it down to the 10 milliliter mark. As far as you can tell just by that measurement, you dispensed exactly 10 milliliters. But if you look at the mass of that water, you realize that you have dispensed a little bit more than what you thought you dispensed. So you actually dispensed 10.04 milliliters rather than 10.00 milliliters. Do the same thing with a 20 milliliter sample, 30 milliliter sample, 40, and 50. And you notice that in each of these cases, we are not reaching the exact amount, which is shown by this black line going straight across. So if any of those points are on that black line, that would mean that we were dispensing exactly the amount that we thought we were. So if we were to run this experiment, determine that there's a little bit of error in the burette measurement, what we could do is apply a correction factor based on this graph. So if we were to measure a specific volume, which would end up being, say, right here, okay, we would think we would measure this volume on the line, but we actually measure this volume here based on the mass. So instead of weighing that particular sample, if we just look on this graph, we would realize that we're measuring a little bit less than what we think we are, and that might end up being something like 29.43 milliliters. Okay, so again, this is a systematic error, not quite dispensing what we think we are, but we're able to find it and correct it. Now, those types of errors, the systematic errors, are going to affect accuracy. So if you're always measuring a pH that's high, that's going to affect your accuracy. If you're dispensing volume of water or titrant from a burette that is sometimes high or sometimes low, then that's going to affect your accuracy. Now, random errors or indeterminate errors, they affect precision, and they arise from the limitations on our ability to make measurements. So if we were to read a scale, we saw this example before, right? So what is the reading up there? Some of you may read it as 58.1, some of you may read it as 58.2, some of you may read it as 58.3. None of you are necessarily wrong, but each of you are going to have a bias in the way you measure that particular value. And that's just a random error that you can't find and you can't correct for it. It's just the way that you will do that measurement. Okay, so reading a scale is an example of a random error. If you are taking a sample from a river, you're clearly not going to be able to take the whole river and do an analysis on it, so you have to take some samples. Now, the samples may not be the same everywhere. There may be different minerals in different areas of it. There may be different oxygen concentrations. There may be different nutrients or microorganisms. So each part of that river may be a little bit different. So taking samples is going to result in a random error. So the only way to get rid of that would be to sample the whole river, but you clearly can't do it. So like systematic error, random error can both be positive or negative, can't be corrected or eliminated. The only way to deal with it is through statistics. Another source of random error is from analytical equipment or instrumentation. So these are electrical instruments that have electrical current running through them, and even if there is no sample running through it, you could get a plot that looks something like this one here on the bottom, where there is a positive and negative signal, albeit a pretty low positive or negative signal, but this is also an example of random error. So one way to deal with it, again, was statistics, but in order to do statistics, you would have to take several measurements of the same sample. So typically you would take at least 3 readings of the same sample, and if you take a few more, your statistics ends up being better, but there is a point of diminishing returns. It's not necessarily a lot better if you take 20 or 50 measurements, for example. Okay, so there's a difference between error and uncertainty, and I've kind of used them both up until now. So error is the difference between a single measurement or result and the expected value. So for example, in the iron experiment, you have to determine the amount of iron in a tablet. The tablet's supposed to have 18 milligrams of iron, but maybe you measure 17 milligrams of iron. So the difference between the 17 and the 18 is going to be a measurement of error. Okay, and again, that result can be affected by both determinant or systematic and indeterminate or random error. Uncertainty expresses the range of possible values for a measurement or result. So it accounts for all errors, whether it's systematic or random. So this is when you would see a plus or minus value after a measurement. So you can only get uncertainties if you've taken more than one measurement, because taking more than one measurement is going to give you that range. It's going to allow you to do some statistics, which will give you that plus or minus value at the end, which we will talk about in later chapters. Okay, so in terms of uncertainty, there are 2 different types of uncertainty: absolute uncertainty and relative uncertainty. Absolute uncertainty is expressed in the same units as the measurement itself. Okay, so if the measurement is in milliliters, like a burette, an absolute uncertainty will also have units of milliliters. So for example, the burettes you use in lab have a absolute uncertainty of plus or minus 0.2 milliliters. So if you were to take any measurement, so for example 20.03 milliliters, you can tack on a plus or minus 0.02 milliliters on the end of that, because that is the uncertainty in that measurement itself. So it could be 20.01 milliliters, it could be 20.05 milliliters. Relative uncertainty compares the size of the absolute uncertainty to its measurement. So it's not going to be in the same units as your actual measurement. So a burette reading of 23.88 milliliters has a relative uncertainty of 0.001 or 0.1%. Okay, so its absolute uncertainty is going to be plus or minus 0.02 milliliters, right? Because this is in units of milliliters. This is also in units of milliliters, so there's an absolute uncertainty. But a relative uncertainty is either a number itself, but more commonly written as a percent. So how do we determine relative uncertainty? That's quite simple. So we have the absolute uncertainty over the magnitude of the measurement. So if we have an example where we have a burette reading of 12.35 milliliters, and the uncertainty, or the absolute uncertainty in the burette, is 0.02 milliliters, what is the relative uncertainty of that measurement? Again, so here's how we could write it. 12.35 plus or minus 0.02 milliliters. To figure out relative uncertainty, we have our absolute uncertainty, which is 0.02. The magnitude of our measurement is 12.35 milliliters. So if we just take that division, do the calculation, we get a relative uncertainty of 0.0016, and we can round that to one significant figure. We'll talk a little bit later on about why an uncertainty is only going to have one significant figure. It doesn't matter as much for relative uncertainty, but when we talk about absolute uncertainty, it should only have one significant figure. So there's our relative uncertainty, 0.002. If we want a percent relative uncertainty, then we just multiply it by 100%, and our percent relative uncertainty is 0.2%. That's all I have for this chapter. In the next chapter, we'll talk a little bit more about uncertainty and how uncertainty can propagate throughout a calculation or through a method.